Hello everyone, my name is Balaji Olawale Quadri, a fresh graduate from Department of Biochemistry, Ladeke Akintola University of Technology, Ogumosho, Nigeria. Before I begin my presentation today, I would like to thank Beyond Science Initiative for this opportunity given to present this research work today. So for today, I will be presenting um, on the topic, the structure-based computational design of novel covalent binders for the treatment of sickle cell disease. Sickle cell disease is a blood disorder and hemoglobinopathy, which is also the most prevalent inherited monogenic disorder that accounts for about 70% of cases worldwide. It is characterized by a mutation in the HBP gene where glutamic acid is replaced with valine. This replacement with valine causes hydrophobic interaction between valine and other amino acids on the hemoglobin chain, such as leucine, alanine, and phenylalanine. This hydrophobic interaction causes the polymerization of the hemoglobin into a rod-like shape. This formation of the rod-like polymer causes an alteration in the shape of the regular biconcave red blood cell to form the characteristic sickle shape of the red blood cell. The sickle red blood cells are unfit for transport function because HBS has low oxygen affinity, which is often worsened in the presence of products of enzyme bisphosphoglycerate mutase and sphingokinase. Also, sickle red blood cell experience abnormal cell homeostasis dehydration, which worsens HBS polymerization, oxidative stress, and nitric oxide synthase inhibition as a result of activities of both accumulated heme in cytosol and NOS competitors such as ADMA. The molecular and cellular mechanism which are compromised and thus causing painful psychological morbidity such as chronic anemia, chronic and acute pain, weakness and dizziness, vasoconstriction, stroke, organ failure, and early mortalities in patients. Though there has been several successful means of management of the disease such as hematopoietic stem transfer, genetic engineering, and the recent uh, development of voxelotor, which has probably been the most successful therapeutic candidate in the treatment of this disease. However, there are still some setbacks in the fact that voxelotor has not been approved for use in children under the age of 4 years and its long-term side effects are yet unknown. So this study aims to identify compound with better therapeutic efficacy compared to voxelotor. In this study, we used a similarity-based search on the PubChem database using voxelotor as our primary query, and we generated a library of 173 compounds from the PubChem web server. We also used the data warehouse software to generate random molecules, and were able to compile a library of total 74,213 compounds from approved drugs and natural compounds. These compounds were altogether filtered to identify compound with aldehyde warhead that could form covalent interaction with the reactive residue on the hemoglobin chain. We then conducted the covalent docking and the best compound were screened for their ADMET properties where we identify their pharmacokinetic and their pharmacodynamic properties. The three best compounds from ADMET screening were then selected and carried forward into molecular dynamics simulation to better understand the integrity and stability of their complex, after which molecular mechanism calculation were conducted. This is a diagram showing the selected compound and the standard compound voxelotor. This table here shows the interaction profile of the three selected compound and voxelotor. And from this table, it can be deduced that the three compounds outperform voxelotor in terms of their binding affinity and the nature of the interactions formed. The several interactions recorded were covalent interaction, hydrogen bond interaction, hydrophobic interaction, polar bond charged, polar positive charged bond, and polar negatively charged bond, and salt bridge, which was recorded for compound 166. Salt bridge are known to be the strongest non-covalent intermolecular interaction, and compound 166 also recorded two pi cation bond and one pi cation bond for compound 2335. Pi cation bond are known to be strong intermolecular interaction, stronger than hydrogen bond interaction but not as strong as salt bridges, but they are known to be resistant to effect of solvation and pH. These are the interaction profile of the compound with the protein target in 2D. This is the interaction for voxylator for compound 166, compound 2301, and compound 2335. We also conducted post-docking MMG-BSD calculation to calculate the free energy release by the selected compound and voxylator, and it was noted that voxylator and compound 166 had lower energy release compared to compound 2301 and compound 2335. And it can be seen here that compound 2335 had the highest energy release in the formation of their complex with the protein target. To account for the pharmacodynamic and pharmacokinetic properties of the selected compound in comparison to voxylator, we conducted ADME and toxicity analysis using the iDrug web server, and we used a total of 21 descriptors to account for the absorption, the distribution, the metabolism, excretion, and toxicity properties of this selected compound. And in several occasions, the selected compound was seen to perform better or even on the same level as voxylator, except in few cases, such as the case of compound 2301, where it had a high probability of inhibiting the P-glycoprotein, and in the case of low human intestinal absorption rate of compound 166. We also have the case of a high probability of inhibition of CYP1A2 by compound 2335 and a low clearance rate of compound 166. Aside these few defects, the three compounds have been seen to have a good ADME and toxicity profile, and they also lack in places where voxylator lacks, which can be ignored to some extent. Molecular dynamics simulation provides additional insight into the stability of the docked complex, the dynamic of the ligand-receptor interaction, and the impact of various environmental factors such as solvent molecule and temperature on the system. To this end, we subjected the selected compound to 1 microseconds of MD simulation, aiming to understand their dynamic nature in comparison with the standard drug. And we use major metrics such as the protein ligand RMSD, the ROG, the RMSF, and the protein ligand H bond. It can be seen from the RMSD graph here that compound 166 and voxylator tend to be the most stable tend to have the most stable complex with the protein target over the simulation time, with compound 166 being exceptional as it has a nearly stable complex throughout the simulation period, with compound 2301 having the least stable complex with several spikes at several intervals during the simulation period. The RMSF metric was employed to measure the average displacement of every amino acid residue in the receptor relative to the reference structure. And during the course of simulation, it can be seen from the RMSF graph that all amino acid residue, all 141 amino acid residue in the protein structure maintain a relative stability during the simulation time, and it can be seen that compound 2301 and compound 2335 complex had the highest average RMSF during the simulation time, and compound 166 had the least average RMSF, which can be concluded that compound 166 had the highest stability of all the complex, even better than the standard voxylator. The radius of gyration was employed to monitor the conformational changes of the protein structure over the course of the simulation. It has been established that if the radius of gyration decreases during a simulation, it indicates that the protein structure has become more compact, while an increase in the radius of gyration indicates that the structure is becoming more extended. According to the REG value for voxylator for compound 2301 and compound 166, it is evident that they have maintained a strong compactness throughout the simulation period. However, for the compound 2335 complex, it had the highest ROG value, and it can be deduced from this that it was the least compact of the three selected compounds throughout the simulation time. It is also known generally that the presence of a large number of hydrogen bonds between the protein ligand complex denotes a strong binding interaction in the complex. And from this protein ligand H bond, it can be seen that compound 166 is the only protein ligand complex with the highest average intermolecular H bond, and thus we can infer that it is the most stable complex compared to others. And it can be seen from this graph that compound voxylator has the least number of hydrogen bonds, which can be referred back to its molecular docking result, where it had a single hydrogen bond with water molecule and a single uh, covalent interaction. We also conducted post-MD MMPBSA to have an insight into the energy contributed by individual amino acid to the bond in each of the complexes, and the free energy decomposition graph is as seen below. In this study, we used several computational techniques such as covalent docking, Padmet profiling, molecular dynamics simulation, and MMPBSA calculation in a bid to identify compound with better therapeutic properties compared to voxylator. In our efforts in this study, we were able to identify three promising compounds: compound 166, compound 2335, and compound 2301, which had better binding affinity and interaction profile compared to the standard voxylator. However, compound 166 stood out to be the best of the set, backed up by results of molecular dynamics simulation and MMPBSA calculation. Thereby, we conclude that this compound could help to advance translational research in clinical trials and drug development against sickle cell disease. I would like to use this moment to acknowledge my team members that were involved in the success of this research work, and I would also like to take this moment to appreciate Beyond Science Initiative for the opportunity given to be able to present this work at this conference. Thank you for listening. Have a wonderful day.